Our last speaker is Addison Quinones. He's a fourth-year medical student at Mount Sinai. He did his undergraduate at Johns Hopkins, where he majored in neuroscience. He grew up in York, Pennsylvania. Following graduation from undergrad, spent a year as a medical assistant for Mohs micrographic surgery. Then he came to medical school here at Sinai, where he's a fourth-year medical student. He's worked with numerous attendings on a variety of projects, including traumatic brain injury and neurosurgical oncology. He's written book chapters, authored many peer-reviewed journal articles, and posted presentations in his free time. He enjoys football, weightlifting, travel, snowboarding, and salsa dancing. Looking forward to your presentation, Addison.
Great, thank you for the introduction, Dr. Kellner. I'm going to go ahead and share my screen. All right, you guys are seeing this okay, I'm assuming. Okay, so thank you for the introduction, and thank you to the neurosurgery department here at Mount Sinai for allowing me to present this research with you today. So today I'm going to be talking about a study I worked on while I was a summer research fellow at MSK, and this was actually published in JNS last November. And the topic is regarding the use of functional MRI to demonstrate translocation of language function in patients with brain tumors. Okay, so first I just want to provide a brief overview of functional MRI. So we know it measures changes in blood flow due to brain activity. This is what's known as the bold signal. And to measure these changes in activity, they need to induce stimuli in the patient. These are usually done through visual, auditory, or tactile means. And there are typically two conditions when performing an fMRI. There's an experimental condition when they're performing an active task, and then there's the control condition when they're at rest. And when you alternate these conditions back and forth, this is what's known as a block design. This is what we used in our study. So if we look in the bottom right, we can see this diagram kind of explaining how a block design works. So the A is the control condition, the B is the experimental condition. And you can see when it rotates between A and B, the bold response increases, showing that there actually is some increase of brain activity. And lastly, we use functional MRI for localization of patient-specific eloquent areas. This can be things such as language areas, which is what we used in our study, but also auditory activation or motor activation. To provide some background on this study, when I mention cortical reorganization or translocation, I'm referring to a shift of neurological function from one area of the brain to another. Specifically, when I say translocation, I'm talking about contralateral translocation from one hemisphere to the other. Also, reorganization has been shown to occur in various pathologies, including AVMs, strokes, epilepsy, and brain tumors. And we also know that the majority of people have left language dominance, but it has been reported that up to 15% of individuals have right dominance. This is mostly in people who are left-handed. And just to give some info on prior studies on this topic, they mostly look at a single time point, and there's also limited research into factors that are associated with this translocation. So these are some of the questions that we were trying to address with this study. So the objective was to investigate factors which affect translocation of language function to the contralateral hemisphere in patients with brain tumors. This was a retrospective study. Our patient population consisted of those patients diagnosed with a primary brain tumor who had two separate fMRI scans between 2007 and 2021 at MSK. So all these patients had an initial diagnosis, a first scan followed by surgical resection, unfortunately followed by tumor recurrence with a repeat scan for further surgical feasibility. And lastly, they all had to have completed a language task paradigm for their functional fMRI. All right, so for our task-based fMRI paradigm, like I mentioned, we used the block design. This consisted of 8 cycles, rotating between a 20-second active task followed by 30 seconds of rest. We used the phonemic fluency task. So essentially, the patients would be laying in the scanner. There would be a screen above them, and they would display a letter on the screen. And whatever letter that was presented, they needed to mentally generate words that started with that letter. So for an example, C, cat, D, dog, and so on. An important point to note is that these were visual cues, and we wanted the patients to have mental generation of these words. We didn't want them to actually mouth the words or speak the words, because then that would activate the motor face area. So that could confound some of the results in fMRI. Just to give an overview of our workflow, so we used AFNI software. So we started out with an anatomical image, and then we overlaid a correlation map of all the brain activity that was found during the fMRI. And then we had to raise the threshold of activation to only select for our regions of interest. In this case, we are focusing on Broca's area. And then over here, we see that we are actually, this is pretty much a confirmation that we are actually measuring correct activation, because we see Broca's area here on the patient's left. And then in this panel on the right, the black tracing in the background shows the patient's bold response, and the red brackets show when the experimental condition is in effect. So we see that the bold response actually fluctuates up and down with the experimental condition. And we compare that to the right side, which we refer to as Broca's area homologue. And there is no correlation between the patient's bold response and the actual time series. So for how did we determine the language dominance in patients, we use what's called the laterality index. As I mentioned, we use Broca's area on the left hemisphere, and then Broca's area homologue, which is in the right hemisphere, as our regions of interest. We use this equation for the laterality index, where V represents the number of voxels activated in the specific region of interest, and A represents the weighted activation. So we took the number of voxels times the weighted activation on the left, and then subtracted the number of voxels times the weighted activation on the right. And we use this ratio to determine our LI, or laterality index. So this ranges from -1 to 1, -1 indicating right dominance, +1 indicating left dominance, and any laterality index in between -0.2 to +0.2 we consider to be codominant. So now moving on to our results, we just have our patient demographics for our patient sample. We had 33 patients matching our inclusion criteria with similar numbers of males and females. Also, we had heavily right-handed sample, which is expected in the general population. The mean age during these scans was 47 years old and 50 years old for fMRI1 versus fMRI2 respectively. And the majority of our tumors did involve the frontal lobe, and tumor pathology consisted of low-grade gliomas, anaplastic gliomas, and glioblastomas. So looking at the laterality results, we actually were able to stratify patients into two groups based on changes in their laterality index from fMRI1 to fMRI2. So the constant group consisted of subjects whose laterality index did not change dominance in between the time interval. So at both scans, they were left dominant, or they were codominant, or they were right dominant at both scans. The reorganized group consisted of patients who began as either left dominant or right dominant at the first scan, and then this dominance shifted at the second scan. So they started out as left, then switched to codominant or right, and then, or they started out as right and switched to codominant or left. We actually found that 9 of the 33 patients, so 27%, underwent contralateral reorganization of language function. The other 24 had constant laterality. 8 of those 9 patients exhibited a shift from left dominance to either codominance or right dominance. And we actually did have 1 patient who had native right dominance that shifted to left dominance. Also, just looking at some comparisons between groups, the percentages of male to female, handedness, and age were similar between these. And both groups included low-grade gliomas, anaplastic gliomas, and GBMs. And we're going to discuss more of the clinical characteristics on the next slide. But for now, I want to look at this graph on the right. So for this, we're looking at the laterality of the constant groups on the left at fMRI1, fMRI2, and the reorganized groups at fMRI1, fMRI2. So when we look at the constant groups, we see that there was no statistical significant difference between these scans in the constant group. But then when we look at the reorganized, there was a difference between them during the scans. And if we also look at the between group comparisons at the constant fMRI1, constant fMRI2 and reorganized fMRI2, we see that there was also a difference between those groups. So this kind of indicates that there actually was some type of translocation of language function in these patients. Okay, so now moving on to some clinical characteristics. We reviewed the patient's clinical data, and it showed that initially 50% of the constant group had aphasia prior to the first fMRI, which increased to 54% after the second scan. However, the reorganized cohort started at a similar rate, 56% aphasia, but this actually decreased to 22% after the second scan. So while the number of patients is too small to generate meaningful statistical tests, it seems as though there may be some association between the conservation of language function and patients with translocation. And as for tumor pathology, the reorganized group had a longer time interval between scans with low-grade gliomas, 105 months versus almost 35 months. So this may indicate some element of a time effect in these patients. And lastly, the reorganized cohort had a greater proportion of tumor invasion into eloquent areas, more specifically Broca's area. So all 8 of the patients with left dominant to co-dominant or right dominant shift had tumors involving Broca's area at the second scan. And in the figure on the right, this is just, this provides 2 illustrative cases. So the top row shows a patient from our reorganized cohort. We see at the first scan, Broca's area activation right here. And then if you look at the second scan, we see that there's no longer Broca's area activation, but there is Broca's homolog activation. And then in the bottom row, we see a constant patient where they had Broca's activation in both the first and second scans. So for the conclusions from this study, it provides further evidence that language translocation occurs in these patients with brain tumors. Also, tumor involvement of Broca's area is associated with translocation, and that translocation in low-grade gliomas is associated with a longer time interval. And lastly, that this translocation may be associated with some decreased aphasia. So as for the significance in this study, it shows that functional imaging may be able to assist with localization of eloquent areas, specifically in those patients who are not amenable to awake mapping during resection. Also, patients with prior surgery or treatment have distorted brain architecture, so it could help in those cases as well. And lastly, if the patient has undergone true translocation, awake mapping might be performed in the wrong areas, so it can give a clue as to where to find these eloquent areas prior to surgery. And we know that extent of resection can influence survival in these patients. So if we're able to better localize these areas, then hopefully we can get a greater extent of resection. So thank you for my acknowledgments. I'd really like to thank my PI, Dr. Haladny, also my other co-authors, and also the Mount Sinai Department of Neurosurgery here. I've been working with a lot of people since my first year in medical school, so I appreciate all of the support and all the guidance that you've guided me over the years. Thank you, and I'm happy to take your questions.
Great. Thank you very much, Addison. Any comments from our tumor faculty?
Sure, yeah, this is Ray Young, one of the tumor surgeons here at Mount Sinai. Great talk, really interesting findings here in terms of the correlations between lower grade tumors and time interval. I found that really interesting. Do you have any comment about any correlations between the intraoperative mapping results? I assume that each of these patients was mapped intraoperatively. Were there any differences seen between those that on the second surgery, for example, who underwent who had some evidence of translocation, whether there were different findings during mapping?
Yeah, so we did look into this. We couldn't really have like statistical comparisons for that question when regarding intraoperative cortical stimulation versus the fMRI, because for a good amount of these patients, specifically the ones who had reorganization, they were not able to achieve speech arrest when they were doing the intraoperative mapping. And we think that's because of the translocation, but they did try to achieve that in most of these patients.
Yeah, I agree. Sometimes it can be very difficult to consistently know whether you're just not really hitting the area, it's not there, versus technical issues with the speech arrest induction with the cortical stimulation.
Can I ask a question?
Yeah, go ahead.
Were you able to form or test or begin to test any hypotheses as to mechanism of translocation in the patients who you observed to translocate? For example, were you able to assess for the degree of perhaps bilateral language representation on the first fMRI in some patients? And is it possible that maybe those patients who had some level of bilateral representation were able to activate the contralateral side when the ipsilateral side was was affected?
Yeah, so that's our hypothesis as well. We think that those patients who did experience this shift, so to say, had greater codominance at baseline. And then like when the native areas were damaged, then the connections were strengthened. But we didn't have any specific tests for looking at this. It was really just looking at the initial laterality index. So if there was any degree of codominance, then we were able to see that during the initial fMRI.
And was that, maybe I missed it, but was that initial degree of codominance correlated at all with translocation?
No, we didn't find. Sorry, I'm just trying to think. We didn't see any degree of codominance that was associated initially, at least.
Is that answering your question, Ben?
It does. Yeah. Yeah. I would be interested in the mechanism of this translocation and whether there's a protocol that can get at mechanism over time. It's a very interesting phenomenon.
Seems like you have some information about rate of translocation, and it correlated to the degree of translocation correlated to the length of time between the scans. Is there any way you can calculate a rate of translocation?
When you speak about rate, are you mean in terms of like the speed, like how like when it occurs, or in terms of just?
Yeah, how long it takes for translocation to occur?
Yeah, so for the interval timing that we have, this was really just based on how long it was until the patient had recurrence, and so then our time interval was based on when they had the first scan and when they had the second scan. So that is all the timing information that we have, but we don't really have any information into when the actual translocation might have occurred because we weren't serially scanning them repeatedly. So we just have those 2 time points.
Dan, comments?
So to me, translocation across hemispheres represents a sort of extreme example. There was a paper, from Eddie Chang's lab at UCSF last year that showed there's no relationship between resecting anatomical Broca's area and Broca's aphasia postoperatively in tumor patients. And so, I'm curious, did you look at translocation from Broca's area ipsilaterally to different regions? And what they found was sensory motor cortex, supramarginal gyri, much more associated with postoperative Broca's aphasia. And I know you weren't able to look at the actual intraoperative data, but did you look at ipsilateral migration from anatomical Broca's area to other regions ipsilaterally?
Yeah, so we actually in this specific study we did not, but there is a follow-up study from our lab. I was not a part of this, but they actually used multiple indices. So they looked at like activation of Broca's area and Wernicke's area. But specifically for this study, we did not look at those areas because we found that with phonemic fluency, the task that we have, that Broca's area activation was the most specific for laterality dominance. So I can't really provide an answer for that in return in terms of this study.
This was a really great session we had today. One of the things I liked a lot about it was that at least for the Sinai students, you guys are working on each other's papers. I love to see that. I think that Addison, you worked on one of the other papers and vice versa. That's a really nice thing. The other thing that is really fascinating is the growth of neurosurgery into understanding how the mind works. And I see that progressing. You can call it functional neurosurgery, but we're moving strongly in this direction, which I think is really fascinating. So thank you all for your efforts today.
Thanks, everybody. Thanks for 4 excellent talks. All right. Have a great day.